Assalamu alaikum everyone. Today we will discuss about reverse transcriptase polymerase chain reaction RT-PCR. In this lecture we will discuss about what is PCR, what is RT-PCR, types of RT-PCR and the procedure of RT-PCR test. So first of all, what is PCR? PCR stands for polymerase chain reaction. It is a process invented by American biochemist Carey Mullis in 1985 while working for the biotech company in California. It is a method used to make rapidly millions of copies of a very small sample of DNA. As we see in this animation that one molecule of DNA divides into two, then four, and finally leads to the million and billion copies of a DNA from one molecule. To understand RT-PCR, it is important to keep in mind the steps of PCR because RT-PCR have the same steps as the PCR have but little bit modification in it. There are three steps of PCR: denaturation, annealing and extension. Denaturation, in which double-stranded DNA are heated to separate the strands. Annealing, in which short DNA molecule called primers bind to the flanking region, the region of the DNA which is adjacent to the 5' of a gene of a target DNA. Extension, in which DNA polymerase extends the 3' of each primer along the template strand. RT-PCR stands for reverse transcriptase polymerase chain reaction. Viruses have the variety of their genetic material in the form of DNA, RNA or mRNA. As we have the very familiar example of coronavirus which have their genetic material in the form of mRNA. RT-PCR uses the RNA as the starting material for nucleic acid amplification. The process is initiated by the enzyme reverse transcriptase which convert viral mRNA into the DNA and then go through with the simple PCR process. RT-PCR, also known as real-time PCR or quantitative PCR, means it describes the quantity of different PCR products using the fluorescence dye such as cybergreen. The dye binds into the minor groove of the double-stranded DNA and does not bind to the single strands. When bound, it increases its fluorescence by up to the 100-folds. During the process, as the target sequence is amplified, cybergreen binds to the each new copy of the double-stranded during the extension step. Procedure of Rt-Pcr test. There are three major steps: denaturation, annealing and extension. Denaturation. High temperature incubation is used to melt double-stranded DNA into the single strands, usually 95 centigrade temperature. Annealing. In this step, temperature is lowered to approximately 5 degrees centigrade below the melting temperature of the primers to promote the primer binding to the template. Extension. The temperature is increased to 75 centigrade, which is optimum for DNA polymerase activity to allow the hybridized primer to be extended. Types of RT-PCR. There are two types of RT-PCR. First is one-step RT-PCR and the second is two-step RT-PCR. One-step RT-PCR, in which we require only one test tube for the whole process, while in the two-step RT-PCR, we require two test tubes for the whole process. Reverse transcription of the RNA template is performed first once and the amplification of cDNA is carried out in a separate tube. Thank you.